And I put this up here in part because it's a painful reminder that I willingly moved away from this place to snowy Boston, but mostly because I think it's a beautiful illustration of the power of technology, what it can do to solve a problem. Back before the 30s, if you wanted to get from the north of San Francisco into the city, you had to take a painful circuitous route around the bay. But when it became possible to span that distance through new bridge technology, now you can get across from the north into the city in less than a minute. So it's a very nice illustration of you match the right technology with the problem and you can have a very big impact on solving that problem. And for vaccines, I think in order to really address some of the large unmet medical needs that have plagued us for quite a while, we're going to need to apply new technologies. And to understand where one could potentially intervene with the technology, I think it's useful to break the vaccine down into its key components. Of course, you need the antigen, whether that's a protein polysaccharide or other type of antigen. You need to have that in the vaccine to elicit the memory immune response. Now, as we've gone away from live organism vaccines to subunit vaccines, whether they're recombinant proteins, polysaccharides, nucleic acids for that matter, we've lost some of the components that were present in the whole organism vaccine, including an immune stimulus. And what we've found often is we need to add these things back. And what I'll show you and what you'll see nicely from the subsequent presentation by Alan, that if you add components to subunit vaccines, particularly those that target the innate immune system and its pathway specifically, you can have a very powerful impact on the immunogenicity of that protein. You often also need to have a delivery system to make sure that both the antigen and the adjuvant or immune stimulus are at the right place at the right time. And this is not only important for potency, but it's also important for safety. Because with adjuvants, particularly those powerful molecules that interact with the innate immune system, we're often walking a tightrope between bad inflammation and good inflammation. And if we can make sure that that inflammation is focused only where it's needed and not wasted elsewhere, we not only have a better potency, but we also have a better safety with the vaccine. So what I'll do in my presentation is give you 3 examples, one from each of these 3 areas where the use of a technology has been helpful in solving a particular problem. For any vaccine program, when you embark on the program, the first critical question is, what do you put in the vaccine? What antigen do you want to target? And for many viral targets, that's usually pretty clear. For bacterial targets where you have many thousands of components in the bacteria, that isn't always clear. And it's a particular problem for meningitis B, where unlike the other serogroups of meningitis where polysaccharide conjugate vaccines have been very effective, you can't really take that approach with MenB because the capsule of the polysaccharide of MenB is a self-antigen, and therefore you'd have difficulty in breaking tolerance, and even if you were successful, you would have the potential for inducing autoimmunity. So people over many decades have tried to figure out ways to make MenB vaccines by taking what's called the Pasteur's principles. You isolate the organism, grow it up, grind it up, purify the main components, and hope that that will induce effective immunity. And while that's effective for inducing potent responses, it's really restricted to only those serogroups from which the vaccine antigens have been identified and purified. And therefore, the real challenge is not how you make the vaccine, but how you make a vaccine that gives you broad immunity across the different serogroups. So back when it became possible to sequence genomes, we partnered with Craig Venter at the Institute for Genome Research to sequence the genome of meningitis B and use that as the starting information for selecting the antigen to put in the vaccine. And to make a long story short, my colleagues in Siena used a screening funnel and by brute force screened as many antigens that were possible to express for their ability to induce bactericidal functional antibody responses, particularly those that were effective across different serogroups. And what we've ended up with out of this funnel is a candidate vaccine that has 5 components. It's 3 separate proteins, 2 of which are fusion proteins that target 5 separate antigens of the organism. And this is a very effective way at inducing potent and broad immunity across multiple serogroups. And this vaccine is now in registration phases in Europe, and it looks very exciting. We also use this platform now as a general starting point for our bacterial programs to use this screening funnel, so-called reverse vaccinology, to give us an unbiased way to select the most appropriate antigens to put in the vaccine. And I think this has the potential to really change the way we embark on discovery programs for any bacterial or even certain viral. Herpes viruses, for example, have so many antigens that you might want to take this so-called reverse vaccinology approach as well. The second area is increasing the potency of a vaccine. And as an example, I'd like to use pandemic influenza because there's a particular problem here. Unlike seasonal flu, people have not been exposed to pandemic influenzas, particularly the avian influenzas. Therefore, there's no baseline immunity. For seasonal flu, by the time we're adolescents, we've had multiple exposures to either clinical or subclinical infections or even vaccinations. And therefore, although we're still susceptible from year to year, we probably have some underlying baseline immunity that prevents us from getting serious disease every year. That isn't true for pandemic flu and H5N1, and therefore we need ways to, 1, make the vaccine go further, and 2, to make the vaccine potent enough and broad enough that you can have protection quickly, hopefully after a single immunization. And to show you the power of using an adjuvant for an H5N1 vaccine, these are data from a phase 1 trial that we conducted where we used the MF59 adjuvant, which is our oil and water emulsion-based adjuvant that has been used widely. It's in licensed influenza vaccine for the elderly in many countries, but has been clinically evaluated in dozens of different targets and is a very effective way at inducing immunity, particularly antibody protection against viruses and bacteria. These data show that if you adjuvant the vaccine either at a 15 or a 7.5 microgram dose, you get much stronger immune responses as measured either by neutralization or by CD4 T cell responses that are far higher than if you get with the unadjuvanted vaccine alone, which is the bottom curve on both of these graphs, where you can see even after 3 immunizations, you barely have what would be predicted to be protective immunity. So clearly, without an adjuvant, this vaccine is going to be minimally effective, if at all. What this shows is, though, there's a potential strategy here for approaching a pandemic situation, and that is, if we could use a vaccine, even if we don't know what to put in the vaccine precisely at this point in time, to give us some level of baseline immunity, at which point, some point in the future, you could give a second immunization and then very rapidly boost up protective immune responses. The problem, of course, with this approach is we don't know what strain is going to be circulating out there in the future. Therefore, the choice of the vaccine you immunize now becomes a guessing game, really. And then secondly, we don't know what the interval is going to be. It could be many years or decades before we actually have a real serious outbreak of pandemic avian influenza. And therefore, is the immunity going to last long enough? Fortunately, we've had the opportunity to investigate this based on a trial that we originally did back in the late 1990s in Europe, where we used an H5N3 vaccine and tested it again with and without MF59. And these are the data that we obtained during the priming phase back in the late 90s. And you can see, as we've seen with many other vaccines, MF59 really does increase the potency of the vaccine as measured by protective antibody responses. But the question is, now 6 to 8 years later, when we recruited these people for a third immunization, in fact, there were 2 priming doses here. So a third immunization was given with a different strain. This time it was an H5N1 influenza vaccine. And we administered the vaccine 6 to 8 years later. So half the individuals had been primed with the vaccine containing MF59, and the other half been primed with the vaccine with no adjuvant. And the results are here. And you can see again quite clearly, if you look at the upper curve, this is homologous neutralization in those individuals that were boosted but had been primed with MF59, compared to the lower curve here, which is those individuals that were primed without MF59. So you get about a 2-log enhancement of potency as measured by neutralization. But more importantly, if you look at the breadth of the antibody responses, we start to see neutralization against a wide variety of different influenza strains, even from different clades, suggesting that so long as you prime with MF59, you can elicit broad immunity, even if there's a mismatch between the priming vaccine and the boosting vaccine, that you really don't see if you have primed only with the unadjuvanted vaccine. The reason for that is it looks like MF59 not only increases the potency of the vaccine, but also increases the breadth of the antibody repertoire that's elicited. And this is data that we generated in collaboration with Hannah Golding at the CDC using phage display to map the B-cell epitopes of the antibody that were generated in these individuals. The upper panel shows an unadjuvanted vaccine, and you can see that the majority of the epitopes that were elicited by the unadjuvanted vaccine reside in the HA2 region of influenza hemagglutinin, and really no measurable responses to neuraminidase. In contrast, those people that were given the vaccine with MF59 also had plenty of responses to HA2, but lots of responses also in the HA1, including the receptor binding domain, as well as responses in neuraminidase. So clearly there's some broadening of the repertoire of the antibody responses with this adjuvant. We also looked at that in the more recent context of the H5N1 vaccine. And using the same approach, we looked at individuals prior to vaccination. And again, you see HA2 specific antibody responses predominate in unvaccinated individuals. But of course, these people have been exposed previously. These are healthy adults who've undoubtedly have had infections or vaccinations prior. In the group that received the vaccine without adjuvant, very similar to the H5, the bulk of the responses are in the HA2. You do see some responses, and they do span this receptor binding domain, but if you compare the width of these bars, you see that the magnitude of those antibody responses, recognizing those epitopes, is very small compared to the ones that predominate in HA2. Again, very similar to the H5, with the adjuvant, you see many, many more epitopes and a lot more frequency of these responses in the adjuvanted group, clearly suggesting that not only potency but quality of the response is increased, which could explain why we have cross-neutralization across clades if you immunize with MF59 versus without MF59. So how is MF59 working? We've known for quite a long time that if you immunize with an antigen with MF59, in this case we were using the herpes GD recombinant protein, that it facilitates uptake of the antigen at the injection site into antigen-presenting cells, and those antigen-presenting cells then migrate to the draining lymph node and present the antigen to T cells and B cells. If you look here at this panel versus that panel, you can see that in the presence of MF59, you see a lot of signal of a fluorescently tagged antigen in these cells, but you don't see it with the unadjuvanted form of the protein, clearly suggesting that by whatever mechanism, it's facilitating uptake of the antigen into the cells. But what we also know is that, first of all, there's no interaction between MF59 and the antigen, unlike with alum. So MF59 is probably not acting as a delivery system directly. We also know that you can separate in time MF59 from the antigen. That is, you can inject the muscle with MF59 and then come in some hours to a day later with the antigen, and you still see the effect. So clearly you can separate the delivery of the antigen from the adjuvant effect. And probably by whatever mechanism that MF59 is facilitating uptake, it's an indirect mechanism, possibly through facilitating phagocytic activity of antigen-presenting cells, as an example. More recently, we've looked at this at a molecular level and tried to understand what is going on in the muscle compartment at the injection site when you immunize with MF59. And this is transcriptome analysis showing that many, many genes are upregulated but when you include adjuvants like MF59 or CpG or alum for that matter. But that if you look at the universe of upregulated genes, you see this red circle represents MF59. The smaller circles represent CpG or alum. And clearly MF59 induces a larger repertoire of upregulated genes than does CpG or alum. There are a lot of genes that are in common, of course, in the middle here. But what we find for MF59, that there are many genes that are upregulated, are those that are involved in cytokine production, chemokine production, suggesting that one potential mechanism is recruiting and activating antigen-presenting cells to the site by some possibly indirect mechanism. What I failed to mention here, in addition, was that we've found that MF59 in vitro does not activate dendritic cells or cells directly. So it's an indirect mechanism of upregulating some constellation of genes, which then downstream affects dendritic cells to activate them. When we looked in the muscles, we found that, as expected, there was a significant infiltration of a variety of different immune cells to the injection site within a period of hours to a few days, and then these were cleared. So based on all of this information, we believe that MF59 is acting by at least two mechanisms, and that is by upregulating a series of genes, including those that facilitate the recruitment and activation of antigen-presenting cells to the site, either directly or indirectly facilitates antigen uptake at the injection site and then migration of those antigen-loaded dendritic cells to the draining lymph node, which ultimately responds in a greater level of priming of antibodies and T cells against the antigen. Based on this information, what we were also interested to see was, since this doesn't seem to have a direct effect on the innate immune system, what would happen if we included molecules that are known to have a direct effect? Is there any synergy or additive effects of a direct versus an indirect immune stimulus? And the answer is you can have quite profound synergistic effects by simply adding in any one of a number of different toll-like receptor agonists. And this is a mouse experiment where we looked at antibodies and T cells against HIV envelope protein, either with MF59 alone or by simply adding in a number of different toll-like receptor agonists. And you can see that you can have orders of magnitude enhancement of antibody responses, as well as quite profound shifting of the profile, the phenotype of the helper T cell response, particularly those that would have a Th1 bias, such as TLR3, 7, and 9 agonists. So given this information, we embarked on a program to mine small molecule libraries for compounds that would have potential adjuvant activity. And we based this on an in vitro screen using cells, and we did a number of different screens, all functional screens, looking at the ability of compounds to elicit a particular response on these cells. Could be cytokine production, could be activation, cell surface marker upregulation, et cetera. And this is actually a plate from one of the screens, and you can see that there are a couple of hits on this plate, a low potency hit as well as a high potency hit. And we screened a couple of million different compounds out of Novartis's and Chiron's libraries of small molecules, and we were surprised to see the very broad repertoire of chemotypes and chemical structures in the chemical space that have the ability to induce these kinds of responses in vitro. And when we took a selected few of these to animal studies, there was a very good correlation between the ability of these compounds to stimulate an in vitro response as measured by a cytokine production, for example, and its ability to adjuvant a vaccine in an animal. And so we have now a number of these compounds that are being developed as vaccine adjuvants and are in the late stages of clinical development, and we're pretty interested to see how these work in larger animals and humans eventually. So the last topic I'd like to touch on is vaccine delivery. And the vaccine platform that is probably most in need of a delivery system is the nucleic acid or DNA vaccine technology. And despite what I may have conveyed in the panel session this morning, I'm a firm believer that DNA vaccines will eventually work. We already know that proof of concept has been established in humans. They're immunogenic, both for antibodies and T-cell responses. And in fact, we have several licensed products in the animal health sphere, including one from Novartis. And so I believe it's just a matter of finding the right technology to enable efficient delivery of DNA vaccines and efficient co-stimulation of the innate immune system to get sufficient potency to make these vaccines viable for humans. So we, over the years, and many other groups, spent a lot of time looking at potential enabling technologies for DNA vaccines. And the one that we took to phase 1 trials was a PLG microparticle-based formulation in an attempt to make it look like a microbe, about a micron in diameter. They're surface adsorbed, the plasmids onto the surface so that you would have an efficient uptake of the particles by antigen-presenting cells, but that the DNA would rapidly come off the surface and you'd have an efficient means to deliver. And based on a lot of in vitro, preclinical animal data, including monkeys, this was a very powerful way of inducing not only potent antibody responses, but also T-cell responses, including CTLs. So through the HIV Clinical Trials Network, we evaluated this formulation in an HIV in healthy individuals with DNAs encoding HIV envelope and HIV GAG, and we used the DNA on PLG as the priming vaccine, followed by a booster with recombinant envelope protein produced in CHO cells, and we engineered these proteins to be trimers. Here's some EM pictures down here, and adjuvanted with MF59. And based on the preclinical monkey data, this was a very powerful combination to get both antibodies and T cells, including CTLs, much higher levels of both antibodies and T cells that you can get with either the DNA vaccine or the protein alone. And the data are shown here. This is the phase 1 trial. And very much as we'd seen with the monkeys, if you prime with DNA and then boost with protein, you get not only higher neutralization, but also higher T-cell responses than you get with the protein alone. We did not have in this trial DNA alone, but based on our monkey data, we would predict that the DNA vaccine, even formulated, is not a very good way of priming neutralization, and you really need to have the protein. But we clearly demonstrated that the DNA prime sets up the individuals for having higher levels of neutralization than if you give just the protein alone. If we look at the phenotype of the T-cell responses, as expected, the DNA vaccine was biasing the helper T-cell response to more of Th1, and these are just 3 different dosing groups of DNA, and you can see that the vast majority of the antigen-specific T-cells in these individuals were secreting interferon gamma, whereas in contrast, you have the converse, where most of the T-cells elicited by the protein-alone vaccine are Th2, like secreting IL-4 and other Th2 cytokines. So this was a very nice recapitulation of the primate data with the DNA prime protein boost. The problem is that even though it was immunogenic, the antibody responses were fairly narrow, and the neutralization was restricted only to those relatively easy to neutralize viruses. So although it's an interesting way to elicit broad immunity, both antibodies and T cells, this is probably not a solution for HIV. So in parallel, we have been working on a different kind of nucleic acid vaccine. It's an RNA-based vaccine that uses the genome of the alpha virus replicons. And this is an RNA virus that has the ability to enter cells and replicate in the cytoplasm by virtue of encoding the replicase machinery for making the RNA and expressing the RNA in the cytoplasm of an infected host cell. This approach has several potential advantages over other nucleic acids, particular naked DNA vaccines, for example. Because of the fact that they're packaged with the viral coat, they have the built-in efficiency of the delivery. Because the RNA does not need to get out of the cytoplasm, you take away one barrier that the DNA needs to get across, and that is the nuclear membrane. And we know that that's a particular limitation of DNA vaccines in non-dividing cells. We also have the potential for amplification because the replicon has been engineered to remove the structural proteins, and you encode the gene of interest, in this case the antigen, rather than the structural proteins of the coat. This is a single cycle replication where you express your antigen, but because you're not expressing the coat proteins, there's no possibility of creating a newly infectious viral particle. And finally, the RNA that is produced here has the potential to be a self-adjuvant. It's known to stimulate TLR3, RIG-I, and other non-TLR agonist pathways in the innate immune system. So it has a number of potential attributes that would make it a very attractive way to take advantage of this particular way of delivering a nucleic acid. And just to show you one piece of data, this is again in the HIV model where we've immunized macaques, again in a prime boost scenario with a V-Syn replicon. These are two different kinds of alpha viruses, the Venezuelan equine encephalitis and an avian Synbis virus. It's a chimeric where we have the replicon from VEE, but the packaged coat from Synbis as the priming vaccine, and again boosted with MF59 adjuvanted envelope protein. And then the animals were challenged with SHIV, and we measured viral replication in these animals. In the unimmunized animals, you see very high levels of viral replication within a few weeks. It's a self-limiting infection that eventually clears by about 2 weeks, but clearly all the animals had very high levels of viremia. In contrast, the immunized animals, only a few of them had any measurable levels of viremia at any point in time after challenge, indicating the power of this prime boost approach with the alpha virus replicons as the nucleic acid prime and the adjuvanted protein as the boost. I don't have on here the data, but the protein alone, as well as the PLG DNA, did give protection, but it was intermediate. It wasn't nearly as good as the alpha virus replicon as the priming vaccine. So in summary, if we are to be able to make inroads into the big unmet medical needs that still exist out there, particularly those targets that might require potent T-cell responses, require broad responses against different serotypes, or potentially therapeutic applications of vaccines, we clearly need to have new enabling technologies in our arsenal or we're not going to be successful. I talked a little bit about reverse vaccinology and the antigen selection process, but other paradigms that we are embarking on in the infancy stages still, particularly for viral vaccines. Not only do you need to choose the right antigen, you need to how to present it to the immune system appropriately. So how to take rational approaches to designing protein-based vaccines that mimic the structures that are on the surface of the virus and therefore induce the protective immune responses is going to be a challenge. And then finally, eventually, we would like to get away from using biological systems altogether and get toward something that we're calling synthetic vaccinology. And again, we are collaborating with Craig Venter to try to take advantage of some of his breakthroughs in large genome size synthesis of DNA so that you could eventually get to a place where you could, even for organisms, have a completely synthetic approach to make the vaccine. So I'll stop there and be happy to answer any questions. Thank you for your attention.
Yes. Microphone's coming.
This is Bill Taylor with Phoenix Inc. Question on your MenB reverse vaccinology. You said you started off with about 600 potential in silico targets and attempted in E. coli and got about a 50% hit rate from that. Had you ever thought about looking at alternate ways to express those to get a higher initial screening?
Yeah, in fact, we played around with DNA vaccines for that purpose because some of the problems there were for the highly transmembrane domain type proteins. They were difficult to express and purify. The DNA vaccine approach worked to a certain extent, but wasn't the solution. So surely there are better ways to express. But we've now streamlined that whole process of selecting. And it isn't completely unbiased anymore. Some of the interesting observations we made along the way were that you can eliminate some of the open reading frames based on their predicted location in the bacteria. So if it's not a surface protein, there isn't much point in generating an antibody response. So we've now figured out ways to triage, and you don't have to express the entire genome to screen.
Other questions? Jeff, in the compound screens where you're looking for immunopotentiators, were you actually specifically looking, or in addition screening for those that would enhance MF59 activity?
No, we only tested those after we had identified them, that they had activity in vitro.
Right.
And many of these compounds have adjuvant activity even unformulated, but when you add them to alum or MF59, you can get very strong potentiation, synergistic effects.
So you have both classes of compounds at this point?
Well, generally speaking, if they're active, they'll be active with MF59 or alum as well.
Okay.
We haven't found any that are only active in that context.
Yeah.
Hi, Jim Matriano, Direct Pharmaceuticals. Question, when you designed the PLGA DNA formulation, was it designed for engulfment or was it for slow release? And was this microsphere based?
Yes, we specifically didn't choose to incorporate the DNA inside the particle, although we evaluated it, because we wanted to have a rapid release of the DNA off the particle. So we chose we looked at different forms of the PLG, those that were fast dissolving versus slow dissolving. There didn't seem to be that much of a difference so long as we appropriately adhered the DNA to the surface in a way that it came off once it got inside the cell. So this was not really a depot by the classical sense.
Hi, Jeff. This is Wouter Latour with Vex Ventures. I was wondering, the small molecule adjuvant that you identified, is that in the public domain, or if not, can you say a little bit more about the composition, its nature?
They are not yet in the public domain, so I can't comment on their structures, but I can say that there are a very wide variety of chemical structures, some of which are known to function through TLR-dependent pathways and others that are not.
Any other questions? Well, let's thank Jeff again.
Thank you.